All right. Today, we are diving deep right into the tiny machinery inside our own cells to get a handle on one of the biggest challenges in medicine, cancer. And it really all boils down to a set of instructions, a master plan that when it gets garbled can have some pretty devastating consequences. So let's get right into it. Here's a question for you. It sounds simple, right? But the answer is unbelievably complex, and it gets right to the heart of what we're talking about today. See, every single healthy cell in your body is playing by a very strict set of rules about when to grow, when to divide, and, this is the most important part, when to stop. So who or what is enforcing these rules?
I want you to think of a healthy cell kind of like a perfectly run factory. It's got a master plan. That's our DNA. And this plan dictates every single move. It makes sure that cells only divide when they're supposed to, which creates this perfect, delicate balance that keeps us healthy. But a plan is just a piece of paper, right? It's useless if there's no one there to read it.
And that brings us to the absolute key players in this story, the gatekeepers of our genes. These are the molecules that are actually reading that DNA instruction manual and deciding which commands to follow and when.
So what are these things called? Well, the official name for these gatekeepers is transcription factors. They're proteins, and you can basically think of them as a biological on/off switch. By latching onto our DNA, they can turn a gene on, letting its information be read, or flip it off, shutting down the flow of information completely.
So here's the bottom line. These transcription factors are the managers of the cell cycle. They're the ones responsible for hitting the go button for cell division, or, just as importantly, hitting the stop button. In a healthy system, this whole process is just beautifully regulated, everything running like clockwork. But what happens when the managers stop following the rules?
And this right here is where things can go terribly wrong. A single mistake, just one mutation, can turn these careful regulators into total rogue agents, and that leads to chaos inside the cell. Let's take a look at exactly how that control can be lost.
Okay, so there are two main ways this whole system can break down. First, the gatekeeper itself, the transcription factor protein, can mutate. It basically gets stuck in the on position and becomes hyperactive. Or second, the gate, that specific spot on the DNA where the factor is supposed to bind, can mutate. This change can make that gate, let's say, stickier, causing the transcription factor to attach way more often than it should.
And here's the thing. It doesn't matter if it's the gatekeeper that's broken or the gate itself. The outcome is exactly the same. The growth signal becomes relentless, it's on all the time, and the cell just starts dividing and dividing without any restraint. This is the very beginning of abnormal cell growth.
So, we've been talking about these tiny molecular mistakes. Let's make this real. How does this actually connect to a disease we all know? Because this isn't just theory. This exact process is a well-known pathway for how cancer develops. Let's take a specific case study, a certain type of breast cancer. In these cancers, the problem kicks off with a protein called EGFR, which stands for epidermal growth factor receptor. The cell just goes haywire and starts making way too many of these receptors, dotting its entire surface with them.
And having all these extra EGFR proteins sets off this dangerous cascade. Suddenly, the cell is just bombarded with growth signals. This triggers a chain reaction of other proteins inside the cell, which then go and activate our gatekeepers, the transcription factors. They get stuck in the on position, permanently turning on the genes for cell division. And that leads to the uncontrolled growth that we recognize as cancer. But, and this is so important, here is the hopeful part of this whole story. By understanding exactly how this control is lost, scientists and doctors can start to figure out how to get it back. It's like the problem itself contains the blueprint for its own solution.
And this is really the key insight, right? By pinpointing the exact breakdown in the system, like that hyperactive EGFR pathway, researchers suddenly have a specific target. If you know exactly which switch is stuck, you can start designing something to unstick it.
And that is exactly what has happened. Modern medicine now has what we call targeted therapies, drugs that are designed to do one very specific job. In this case, they block that EGFR protein, stopping it from ever starting that dangerous chain reaction. It's basically like finding and cutting the power cord to the cancer's growth engine.
So, let's do a super quick recap of what we've covered. The cell's gatekeepers, these transcription factors, are absolutely essential for normal growth. But mutations can mess with either the factors themselves or the DNA they bind to, causing them to go into overdrive. Both of these paths can lead to the uncontrolled growth that we see in cancer.
But, and this is the most important part, understanding these very specific pathways is what allows for the creation of smarter, targeted cancer therapies.
And all of this leaves us with a pretty fascinating final thought. We've seen that cancer, at its very core, is a disease of broken information, a glitch in the cell's master plan. So as our ability to read and understand that plan gets better and better, it poses this ultimate question. Can we one day move beyond just blocking the broken signals and actually learn how to repair them? Could we rewrite the story of the cell itself?